Assalamu alaikum. Today we are going to discuss about cell death. There is some principle like basic principles are the morphologic hallmark of cell death is loss of the nucleus which occurs through nuclear condensation, pyknosis, fragmentation, karyorrhexis and dissolution, karyolysis. The two mechanisms of cell death are necrosis and apoptosis. What is necrosis? Death of large groups of cells followed by acute inflammation due to some underlying pathologic process, never psychologic, physiologic, divided into several types based on gross features. Gross patterns of necrosis, coagulative necrosis, necrotic tissue that remains firm, cell shape and organ structure are preserved by coagulation of proteins, but the nucleus disappears. Characteristic of ischemic infarction of any organ except the brain. Area of infarcted tissue is often wedge shaped, pointing to focus of vascular occlusion and pale. Red infarction arises if blood re-enters a loosely organized tissue, for example pulmonary or testicular infarction. Liquefactive necrosis. Necrotic tissue that becomes liquefied. Enzymatic lysis of cells and protein results in liquefaction. Characteristic of brain infarction. Proteolytic enzymes from microglial cells liquefy the brain. Abscess. Proteolytic enzymes from neutrophils liquefy the tissue. Pancreatitis. Proteolytic enzymes from pancreas liquefy parenchyma. Gangrenous necrosis. Coagulative necrosis that resembles mummified tissue. Dry gangrene. Characteristic of ischemia of lower limb and GI tract. If superimposed infection of dead tissue occurs, then liquefactive necrosis ensures wet gangrene. Number D. Caseous necrosis. Soft and friable necrotic tissue with cottage cheese-like appearance. Combination of coagulative and liquefactive necrosis. Characteristic of granulomatous inflammation due to tuberculosis or fungal infection. Fat necrosis, necrotic adipose tissue with chalky white appearance due to deposition of calcium. Characteristic of trauma to fat, for example breast and pancreatitis mediated damage of peripancreatic fat. Fatty acids released by trauma, for example to breast or lipase, for example pancreatitis, join with calcium through a process called sponification. Sponification is an example of dystrophic calcification in which calcium deposits on dead tissues. In dystrophic calcification, the necrotic tissue acts as a nidus for calcification in the setting of normal serum calcium and phosphate. Dystrophic calcification is distinct from metastatic calcification, in which high serum calcium or phosphate levels lead to calcium deposition in normal tissues, for example, hyperparathyroidism leading to nephrocalcinosis. Fibrinoid necrosis, necrotic tissue to blood vessel wall. Leaking of proteins, including fibrin, into vessel wall results in bright pink staining of the wall microscopically. Characteristic of malignant hypertension and vasculitis. Apoptosis. What is apoptosis? Energy dependent genetically programmed cell death involving single cell or small groups of cells. For example, endometrial shedding during menstrual cycle, removal of cells during embryogenesis, CD8 plus T cell mediated killing of virally infected cells. Morphology. Dying cell shrinks, leading cytoplasm to become more eosinophilic. Nucleus condenses, pycnosis, and fragments karyophytes. Apoptotic bodies fall from the cell and are removed by macrophages. Apoptosis is not followed by inflammation. Apoptosis is mediated by caspases that activate proteases and endonucleases. Proteases break down the cytoskeleton. Endonucleases break down DNA. Caspases are activated by multiple pathways. Intrinsic mitochondrial pathway, cellular injury, DNA damage, or loss of hormonal stimulation leads to inactivation of BCL2. Lack of BCL2 allows cytochrome C to leak from the inner mitochondrial matrix into the cytoplasm and activate caspases. Extrinsic receptor ligand pathway. FAS ligand binds FAS death receptor CD95 on the target cell, activating caspases. For example, negative selection of thymocytes in thymus. Tumor necrosis factor TNF bind TNF receptor on the target cell, activating caspases. Cytotoxic CD8 plus T cell mediated pathway. Perforins secreted by CD8 plus T cell create pores in membrane of target cells. Granzyme from CD8 plus T cell enters pores and activate caspases. CD8 plus T cell killing of virally infected cells is an example. Hope so you liked the video. Please subscribe.